But I'm going to dispense with the rest of my 2-hour speech and turn it over to Dr. John Yu from Cedars-Sinai, who's going to talk to us about his dendritic cell vaccine down there. So, Dr. John Yu.
First of all, I'd like to thank John for his invitation to participate in this conference. It's been great to meet everybody, and John's done a great job in really putting together a very innovative conference, and I really appreciate all the care he's taken into developing that. Also, I'd like to thank Al Mazzella, who over the years has been very supportive of our vaccine programs and funding our trials and helping us get it out to people who need it most. So appreciate his help very much as well. So today I'll be talking to you about the vaccine clinical trials that we've been developing over the last 11 years at Cedars-Sinai Medical Center, and I'll talk a little bit about the rationale for immunotherapy for brain tumors, and talk about really our results and what we're heading toward in the future. So if I can have the next slide. So what are the problems in brain tumor therapy? The survival, although it's been extended with radiation and temozolomide, and now perhaps with Avastin, still remains relatively limited at about 15 months or so. And so clearly begs for a novel way to target this tumor. And I think we're developing the ideas to really have a novel and potent way of addressing these tumors. Also, tumor cells in brain tumors spread within normal brain tissue, and so that makes it all the more difficult to treat. The cancer cells are infiltrating into all the eloquent areas of the brain that control our speech, movement, emotions, intellect, and really everything that makes us human. And so it really eats away at our own humanity, and really that's really one of the most devastating aspects of this disease, and which really makes us want to target this all the more. We know from research that brain tumors suppress the normal immune system, and in fact that's probably the means by which it grows, that our normal immune responses to kill tumor cells whenever they develop has gone awry and is no longer surveying the body as it should. Next slide, please. So the immune status is depressed in glioma patients at several levels of the immune response. Patients with gliomas have defective dendritic cells. Dendritic cells are the most potent antigen-presenting cells of the immune system. It's like a hound dog given the scent of a prisoner's clothes, and it's sort of that presentation of the scent of the prisoner's clothes that a dendritic cell is doing. The hound dog that then goes after that scent would be like a T cell, or the effector cell, the cell that actually does the killing. So both the dendritic cells and the T cells become defective in patients with cancer, not only brain cancers, but other types of cancers as well. And so our overall strategy with our vaccines is to overcome this immune suppression. Next slide. So immunotherapy has been gaining a lot of traction recently. There was an article in Time recently demonstrating some positive results in large clinical trials in prostate cancer and in patients with a type of lymphoma. And so, whereas before, cancer vaccines were always thought of as a non-effective therapy, it's really gaining traction both from an investor standpoint and from a patient care standpoint, that this really may be maybe not the magic bullet, but certainly an effective bullet that can be used in our armamentarium to treat cancers. And this slide just shows some T cells that are surrounding the cancer cell in an electron microscope. Next slide, please. So the history of immunotherapy is a long one. In 1796, Jenner introduced cowpox immunization to cow hands to prevent a smallpox infection. And he actually, this was before the days of the FDA and IRB. And so he took a cow hand in his farm and gave him the cowpox virus, and then made him put him in the environment with smallpox and found that he was immune to the virus. And now we know that smallpox has been eradicated from the world. And so this sort of strategy is what we're trying to encounter with cancers. Now, cancers are different because they start off in the body, but they still are a foreign thing in the body. It's an abnormal thing. So instead of trying to have a body fight off a virus, we're trying to have the body fight off a cancer. In 1876 through 86, Pasteur introduced a weakened chicken cholera bacterium and also vaccinated against rabies. And I worked at the Pasteur Institute in Paris for a year, and they have a great immunology department. This is where I developed my interest and passion for immune strategies for cancer as well as for infectious diseases. In 1954, Salk introduced the live attenuated polio vaccine, which is rid polio from the Western Hemisphere. And in 1982, antibodies were first used to treat a human B-cell lymphoma. And antibodies, as you know, before, again, was thought to be an ineffective therapy. But now, really, with Erbitux, Avastin, and other antibody therapies, it's really taken hold as a very potent way of treating cancers as well. We feel that cancer vaccines is sort of at the early point of antibody therapies now too. In 1988, tumor-associated antigens were identified. These are specific proteins on cancers that are abnormal, that can be targeted by the immune system. And so this type of strategy of using a vaccine for cancer was first really proposed in 1988. Next slide, please. And this is just a picture of a vaccination of a cow hand, of a farm hand with the cowpox vaccine. Next slide, please. So the immune system of glioma patients are suppressed. There's a decreased function of the immune cells. The number of immune cells are decreased, and the number of suppressor cells, or the cells of the immune system that actually suppresses the immune response, are increased. Next slide, please. For antigen presentation, where the scent of a prisoner's clothes is given to a hound, this antigen presentation is very defective in patients with glioblastomas. Normally, the tumor cells themselves present their abnormal tumor proteins to the immune system, but this is done in a very ineffectual way. Tumor-infiltrating antigen cells, or dendritic cells, that are the professional cells that present these antigens, again, are defective in glioma patients as well. Next slide, please. So dendritic cells are a small component of the white blood cell population. They possess the highest capacity to stimulate T cells, or to tell those T cells to go after this abnormal cancer cell. Next slide, please. So this is what a dendritic cell looks like. They have little dendrites that come out of the main part of their body, and these areas of the dendrites are where the antigens are located. Next slide, please. These dendritic cells go into where cancers are, tumor cells. They normally take up the dead tumor cells. They process the proteins of the cells and cut them up into little 9 amino acid sequences, and they put them on what's called an MHC molecule, or a major histocompatibility molecule, on the surface of the cell. Next slide, please. They go to a lymph node nearby. In the case of a brain cancer, the nearest lymph node are the cervical lymph nodes in the neck. They normally travel to the neck, and then once you see those yellow areas, those are the proteins that are being presented to the T cells, which is the red cell there. So they undergo presentation. The T cell receives the signal from the dendritic cell, say, go after this protein. Next slide, please. based on the signals of the dendritic cell, the T cells that, which now recognize that abnormal protein that multiply ad nauseum. Next slide. Then they go out into the circulation from the lymph node and they circulate through the body. And whereas before it was thought that the brain is an immunoprivileged organ, meaning that immune cells couldn't enter the organ, we've demonstrated first in 2001 that after dendritic cell vaccination, these T cells indeed do localize into human brain tumors. Next slide, please. And so once they go into the brain tumors, now they recognize these as abnormal, just like they would recognize a poliovirus in a cell or anything else as abnormal. And so they kill the tumor cells like they would kill cells that are infected with a poliovirus or a rabies virus. Next slide, please. So the rationale for vaccinating patients with dendritic cells that are loaded with these abnormal tumor proteins is to optimize dendritic cell activity and maturation, where they are able to perform their function of presenting to the T cells and allowing the T cells to multiply. We get a large number of dendritic cells outside of the body, outside of the immunosuppressive milieu of the body, and we allow the cells to process these antigens or the tumor proteins in a more normal environment. Next slide, please. Then we take those dendritic cells that are loaded with the proteins and we vaccinate it under the skin like any type of vaccine that you would get for tetanus or anything like that. So in preliminary studies, we demonstrated that rats that are pulsed, that are given vaccines with dendritic cells pulsed with proteins from rat gliomas allowed these animals to live for a prolonged period of time. Next slide. And we showed that in this case. This is what's called the Kaplan-Meier curve. This is the same kind of curve that's shown at medical meetings for patients that undergo clinical trials, where you compare a control group here, the line on the left side with that of a treatment group, the line on the right side. The x-axis has time, so it's from day 0 after the vaccine to day whatever in the future. And the y-axis has the percent survival. So it starts out with 100% survival, and we all know that we're all mortal, and the question is how long we're going to live. And so the question is, can we extend that survival to a more meaningful time? And so whereas the rats that received the brain cancers that were treated with dendritic cells that weren't loaded with the proteins all died within 25 days after the implantation of the tumor, those animals that received the dendritic cell vaccine lived a lot longer. Next slide, please. And so the hypotheses from these initial animal studies were that dendritic cell vaccines would elicit peripheral T cell cytotoxicity. What that simply means is that it would generate enough T cells that would now recognize the brain cancer cells. The T cells would migrate into and kill intracranial tumor cells. Previously, it was thought that, again, the brain is immunoprivileged, the T cells can't enter. And so the third hypothesis was that dendritic cells would have a clinical benefit for patients with brain cancer. Next slide. And so in our first trial that we started in 1999, we took tumors out, patients with brain tumors. We grew them up in test tubes. We isolated the proteins from these tumors, and we added those proteins onto the dendritic cells, and we vaccinated patients 3 times with this treatment. Next slide. So 10 patients with newly diagnosed glioblastoma or anaplastic astrocytoma were treated. Proteins were taken from the surface of the tumor cells, which were grown in culture or petri dishes. So mononuclear cells or white blood cells were taken from the bloodstream just by a blood draw, and then they were made into dendritic cells by placing them in the culture in specific conditions. And the patients received 3 vaccines. Next slide. And so this is the way that we make the dendritic cells. We load them, we grow them in GM-CSF and IL-4, and then we add the proteins onto the cell surface, as you can see there in the slide. we wash the proteins off the tumor cells. We take those tumor proteins and add them to the dendritic cell. Next slide. And so we published the results of this trial in 2001. It was a cover article in Cancer Research. And in this article, we demonstrated that this type of vaccine was safe in patients, didn't have any significant toxicities, that it elicited a peripheral T-cell response, or the T-cells grew in the bloodstream. And in half the patients that we tested, we looked in their brain tumors. There was a significant T-cell infiltration, as you can see in the bottom part of the figure of that cover, where all these T-cells have now entered the brain tumor, killing it, as compared to prior to the vaccine. In the upper figure, there were no T-cells seen in the tumor previously. And then when we compared the survival of these patients to RTOG results in subgroups of glioblastoma patients, we found that the survival was superior to that patient population. Next slide, please. We then went on to another phase 1 trial. This time we took tumor lysate directly from the tumor, meaning that we took the tumor out of the brain and we freeze-thawed the tumor, isolated all the proteins from the tumor rather than growing them in petri dishes and taking the proteins off the surface. And so this prevented us from getting tumor cells that were artifactual or created while growing in petri dish conditions, and rather took the tumor directly, proteins directly from the tumor. Next slide. And so in this slide, in this paper that we published in 2004, we demonstrated that there was a significant immune response in slightly over half the patients that were vaccinated. There was a very specific immune response against specific tumor antigens or proteins, and these included proteins that are shared with melanoma and breast cancers, like HER2, GP100, and MAGE1. And again, memory T cells that now recognize the tumor could be found in the tumor, as can be seen on the right, as compared to prior to the vaccine on the left. Next slide, please. In the survival study of this phase 1 trial, 7 study patients were compared against 51 control patients treated at our same institution. And the 3 years prior to this vaccine, the median survival of the vaccinated patients were 133 weeks versus 30 weeks in those patients that were not treated with vaccine. And this was found to be statistically significant. This was a small cohort of patients, and so this can't really be generalized to a large group of all comers. Next slide. And this is the survival curve. Pretty similar to what we saw in our rat experiments, where the vaccinated individuals lived longer than those individuals that were not vaccinated. Next slide. So the conclusions from this study was that tumor lysate pulse dendritic cells is safe, that this type of vaccine induced a peripheral cytotoxicity in a subset of patients, that these T cells then could migrate into the brain tumors, and that it induced a prolonged survival in recurrent malignant glioma patients. Next slide. One of the things that we found from our phase 1 trials and our phase 2 trials that we published last year in Cancer Research is that those patients within the trial that had an immune response, when we compared those patients that were in the trial that did not have an immune response to the vaccine, those patients that had an immune response lived longer as a group, as you can see on the left, from our phase 2 trial. And when we add patients from all our trials together, again, there was a benefit seen in all the patients that had an immune response as compared to those that did not have an immune response. So this was really scientifically a better control within the same patient population, and this really demonstrated that an immune response really correlated with a clinical response. Next slide, please. And interestingly, not only did those patients that had a response live longer than those that didn't, but the magnitude of the immune response had a direct correlation with the magnitude of the clinical response. So in the x-axis is survival from the date of vaccine, and the y-axis is the magnitude of the immune response. So as the immune response increases, the survival time increases as well. So there's a clear correlation of a biological response to the vaccine with a clinical response to the vaccine. And so this gave us greater hope that by improving our vaccine, that we could improve patients' survival and outcomes significantly. Next slide, please. One of the things that we found from our analysis of our trials is that those patients that received vaccine therapy plus chemotherapy after their vaccine appeared to have a better survival than those patients that either had chemotherapy or vaccine alone. Next slide, please. And so this is one individual of 4 in this trial that had a dramatic response to temozolomide after their vaccination. So they had their surgery and tumor removed in the second figure, and the third figure several months later they developed another tumor in that same area, and when given chemotherapy, that tumor really responded dramatically more than would be expected with temozolomide alone. And so there appeared to be in several instances in this study, and by looking at overall progression-free survival and survival, that there was a synergy seen more than expected by chemotherapy and vaccine when used together. Next slide, please. We also did a trial of intracranial dendritic cell immunotherapy where we put the dendritic cells actually directly into tumors in the brain. This is a trial that's no longer enrolling patients, but it was an interesting trial. And it was designed for patients where we weren't able to remove the tumor surgically, that it was in an eloquent area, right in the motor area, or in a speech area. And so what we did was in patients with recurrent malignant glioma, whose tumors were less than 3 centimeters in diameter, they would undergo stereotactic radiotherapy. It's like a gamma knife radiosurgery therapy in which the tumor is treated with radiation. Then the patients would undergo a craniotomy, and these dendritic cells would be placed directly into the tumor. So we would remove whatever tumor we could that were outside the eloquent area, and then within the eloquent area we would put dendritic cells. 24 patients were treated in cohorts of 3 with an escalation of every 3 patients up to a dose of 128 million cells. Next slide, please. Patients would receive stereotactic radiotherapy with the X-knife. And this therapy is something that we promote with patients as well, in patients that can't undergo surgery. We feel the next best surgical type of option is radiosurgery, where we focus a high degree of radiation right to the tumor. And what this achieves is obliteration of the tumor in the area while sparing most normal neurons. And so, patients would receive stereotactic radiotherapy up to 25 gray over 2 weeks. Next slide, please. Then the patients would have their surgery and get their dendritic cells placed within the tumor 7 days after the vaccine, after the radiosurgery rather. So in this trial, 2 out of 7 patients tested demonstrated a positive immune response against the tumor. 2 out of 6 patients with operative recurrences had a dramatic T-cell infiltration into their tumor. Next slide, please. And this is, again, similar to our vaccine in the skin. You see a dramatic T-cell infiltration into the tumors. Next slide. So the results of this phase 1 trial is that there were no grade 3 or 4 toxicities. And of the 24 patients, the survival was 9.6 weeks to 132 weeks since their immunotherapy, with a median survival of 53 weeks. And so these are patients with tumors in eloquent areas with residual tumors. So the survival looked encouraging given this subset of very difficult patients to treat. Next slide, please. This was the survival curve of that trial. Next slide, please. So the conclusions was that DC immunotherapy used intracranially is safe, that we see the cytotoxicity in 2 out of 7 patients, which was lower than compared to our intradermal vaccinations, and that intracranial T-cell infiltration occurred in about a third of patients. So the peripheral response and the intracranial response was slightly lower than what we would see when we vaccinate within the skin, which may be what one might expect, because it's another layer of the immune response that would be needed. The dendritic cells would then have to take up the tumor cells in the brain and then travel to the lymph nodes and the cervical lymph node in order to generate a response, whereas those vaccinations in the skin would go directly to the lymph nodes from the periphery. Next slide, please. So, as I discussed last night, there's a new notion in cancer therapy called the notion of a cancer stem cell. And previously, cancers were considered to be a stochastic disease, meaning that every cancer cell was identical to every other cancer cell. But the emerging data would suggest that cancer is a hierarchical disease in which there are very few cells at the top that really have that give the cancer the capacity to be virulent and to grow. And the properties that are marked by cancer stem cells are those that are very similar to normal adult stem cells. Those are the property of self-renewal and ability to differentiate or to become every cell of that particular organ. So the idea of self-renewal means that a cancer stem cell can make one copy of itself and one copy of a cell that differentiates or becomes one of the cells of that organ. In the brain, there are 3 cells: the neuron, the astrocyte, and oligodendrocyte. And so we find that in glioblastomas in particular, that there are all 3 of these cell types within the tumor. So instead of a normal neural stem cell making normal neurons and normal astrocytes, a cancer stem cell will make aberrant astrocytes and aberrant neurons that are growing despite signals in the organ that it shouldn't be growing. So just like stem cells in the body, cancer stem cells have a propensity to be very hardy and to withstand all kinds of bad things happening to it, like radiation. Our stem cells have the ability to fight off radiation, to replace mutations that are caused by radiation and to keep the cells growing. They have the expression of genes that allow the cell to spit out the chemotherapies that come into the tumor. And so the stem cells have a particularly hardy means of withstanding all these therapies. So whereas before, as you can see from the bottom line, we were treating probably the daughter cells of the cancer. And the daughter cells are the cells that have differentiated from the stem cell. So these cells have a very limited capacity to grow, and it's really the cancer stem cell that make up 1 to 5% of the cancer, which really gives the cancer its propensity to grow. And so what we've been doing traditionally with chemotherapy and radiation is we've targeted the daughter cells. So those cells, or the 95% of the cancer that actually responds dramatically to chemotherapy and will make a tumor just disappear on an MRI, after the treatment, you'll see a complete response in an MRI because it's really killed most of the daughter cells, but we haven't really addressed the cancer stem cell. And so within 3 to 6 months, we'll see a regrowth of the tumor after a dramatic response, and that's presumably because we've killed off the daughter cell. So our goal moving forward is to really try to target the cancer stem cell, that the emperor of the disease rather than its little minion workers. So if we can target the cancer stem cell as seen in the upper line, then the daughter cells or the differentiated cells or the worker bees should all degenerate because they really don't have the capacity to make tumors. Next slide, please. And so in order to translate into a therapy, we devised an experiment in which we would get dendritic cells from a patient and then load them with antigens or proteins that are isolated from a patient's cancer stem cells. And we've developed methods to actually isolate the cancer stem cells from brain tumors, and this is through a process that's very similar to isolating normal neural stem cells. And once we have the cancer stem cells, we take the proteins from that, load it onto the dendritic cells, and vaccinate patients with it. Next slide, please. So we did this in an animal rat model of a cancer stem cell that we described in the journal Stem Cells, and found that when we place these cancer stem cells into these rats, as you can see in the middle figure, they grow rampantly in the brain, whereas if you take 100-fold of the daughter cells with the main population of the tumor, it really won't grow in these rats. Next slide, please. So in these rats, we vaccinated those rats with dendritic cells that were either loaded with cancer stem cell proteins or just the whole proteins from the tumor. And only when they were loaded with the cancer stem cell tumor proteins would the animals survive for a long period of time. Whereas when they were loaded with just the diminutive proteins from the tumor itself, then those animals would not survive as long. Next slide, please. We also demonstrated that the immune response against those cancer stem cells were quite favorable. Next slide. So this led us to pursue a novel trial. It's called the Phase 1 open-label multi-center study of ICT-121, therapeutic vaccine in patients with newly diagnosed or recurrent malignant glioma. The investigational compound is it's a peptide or a protein from a cancer stem cell, specifically from an antigen protein called CD133, which is isolated on cancer stem cells. Next slide, please. So at our institution, our ongoing trials right now, some of which are close to closing, are a Phase 2 dendritic cell vaccine for all tumors. So this is a vaccine for all the malignant tumors that we've been enrolling for the past 4 years and really about to close in the near future. A Phase 1 trial of the dendritic cell vaccine and gliadel, which is a BCNU impregnated wafer, and a Phase 1 trial of dendritic cell vaccines with 6 different antigens. We're also opening a trial this month with dendritic cell vaccines loaded with treated with imicomod, which is a cream that induces an immune response. It's actually used for genital warts, but it also gives a signal to the dendritic cells to really improve the immune response. So that's what we're looking for. So I'd like to acknowledge our translational research team, above the scientists that do all the work that allow us to translate these novel therapies into clinical trials, and then our research nurses and our clinical nurses below that help us really get these trials and therapies into patients. So thank you very much for your attention. I think I'm ahead of time, so I'm happy to answer some questions. Yes, ma'am.
The amount of protein expressed in a tumor cell correlated with the degree of malignancy?
That's a very good question. In certain cases there are, but it's all based on individual proteins. So for instance, the EGFR or epidermal growth factor, which is a receptor or protein in the cell that gives the cell signals to grow. We found that in glioblastoma, that receptor or protein is amplified. So there are many more copies of that gene than there should be there. And so that's sort of a marker for glioblastoma, that in most cases that protein is highly expressed. But there are a lot of different mutations in cancers, some of which allow more expression of a protein, for instance, so-called oncogenes, that proteins that induce cancers. And sometimes they actually inhibit the expression of some tumor suppressor genes, those genes that actually suppress tumors from growing. So it's a combination of mutations of proteins, too many proteins that help cells to grow, and less proteins that actually help the cell to stop growing. So all those together will induce cells to grow. Some proteins that are highly expressed on tumors make it more susceptible to attack by T cells. So T cells recognize individual proteins on tumors. So if that protein is being made a lot by that cancer, then that cancer will become more susceptible to killing by the T cells. So it actually helps the immune response in certain ways to have a lot of that protein. Yes, sir.
Are there any treatments that would enhance the effectiveness of the immune response that could be added on top of the vaccine?
Sure, that's a very good question. And so one of the things we're looking at is imicomod, this cream for genital warts. It provides what's called the danger signal as a cream where the vaccine is given. So we know that certain signals during an immune response alert other cells to come in and bolster that immune response, and those are called danger signals. And this happens to be a danger signal called TLR7, which is a toll-like receptor 7. It essentially gives a signal to the immune system that there's some danger in the body. And so others have shown in other vaccines that this signal along with the vaccine will amplify the effects of the vaccine. So if thought intelligently and rational, and there's a rational strategy for improving the immune response, then you know, two separate types of therapies can provide synergy in the immune response. So I think that's sort of the next wave of vaccines, to try to synergize things as much as possible. And as I showed just briefly, there is some synergy of chemotherapy with immunotherapy as well. And that really depends on the timing of when those are given, and those data are coming out more and more to help us try to figure out how to best provide vaccine therapies and chemotherapies together.